Hello students, welcome back to the biology class, chapter 6, Molecular Basis of Inheritance, part 3, the search for the genetic material. Even though the discovery of nuclein by Frederick Michaud and the proposition for principles of inheritance by Gregory Mendel were almost at the same time, by 1926, the question to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most of the cell. But the question of what molecule, that DNA, RNA or protein, was actually the genetic material had not been answered. In 1928, one scientist, Frederick Griffith, through his transforming principle, he conducted a series of experiments on the bacteria. We'll discuss later. In 1928, Frederick Griffith, in a series of experiments with the Streptococcus pneumonia bacteria, because the disease pneumonia is caused by the coccus bacteria, a chain of bacteria, Streptococcus bacteria, witnessed a miraculous transformation in the bacteria. During the course of the experiment, a living organism had changed in physical form. When Streptococcus pneumonia bacteria are cultured in a culture plate, in that some produces smooth shiny colonies or S bacteria, and they are the polysaccharide mucus contains, so they are the capsulated bacteria and they are virulent. Some of them are R bacteria, rough colonies, R bacteria does not contains the polysaccharide cord, so they are the non-capsulated and they are the non-virulent. Capsulated means the cell wall of the bacteria, and virulent means they are the pathogenic, they are disease causing bacteria. S bacteria are causing the diseases, R bacteria does not causes the diseases. See the differences between S strain and R strain bacteria. S strain bacteria are smooth shining colonies, they are the contains the capsulated and also they are the virulent. R strain bacteria are the rough colonies, they are non-capsulated and they are the non-virulent, non-pathogenic. Now we'll discuss about a small experiment. The mice experimental organism infected with the S strain, means they are causing the disease pneumonia and die due to the infection. But mice infected with the R strain do not develop pneumonia disease. S strain injected into mice died. R strain injected into mice live. Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat killed S strain and live R bacteria, the mice died. Moreover, he recovered the living S bacteria from the dead mice. Listen, S strain heat killed bacteria injected into the mice live. S strain heat killed injected with the R strain bacteria. The mice died. What is the reason for that? He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle, transformation, is transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Transformation is a type of reproduction is present in the bacteria. Transfer of genetic material from the dead organisms to the living organism. Dead cells to the dead donor cell to the living recipient cell. Here it is, a dead cell is the S strain and the living recipient cell is the R strain. They are receiving. Because of that reason, mice died. But which is the genetic material? It may be DNA or RNA or it is a protein. But the Frederick Griffith is unable to prove that which is the genetic material or which is the biochemical is present in the transformation. So after some years, in 1944, the work of Oswald Avery, Colin MacLeod and Maclean McCarthy, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith experiment. See that one, Frederick Griffith, it is 1928, Oswald Avery 1944. After a long time, they conducting the experiments, which is their find to discover that which is the genetic material. They started the different types of the experiments, work on the DNA, RNA and proteins. After that, they purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cells to see which ones could transform the live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzyme protease and RNA digesting enzyme RNase did not affect transformation. So they discovered that protease enzyme, RNA digesting enzyme RNase did not affect the transformation. So the transforming substance was not a protein or RNA. Then which digestion with the DNase did DNase enzyme is a DNA digesting enzyme did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. According to the Oswald Avery and his friends discovered that DNA is the hereditary material. Thank you.